All right, ladies and gentlemen, well, welcome to section 48.3. Let's let the action potential begin. I'm sorry, that's a G-rated show here. All right, so the deal we've got, we have these voltage-gated ion channels, these potassium and sodium ion channels. And what can happen is when stimulated, those channels can, well, open up. Now, certain stimuli will cause the potassium channels to open. So, if we have the environment inside, here's the potassium, here's the sodium, we have the environment outside, we know it's negative here and positive here. Well, if we have positive potassium ions leaking out through this channel, as a result of that, well, it's going to become more negative on the inside, and that leads to what's referred to here as hyperpolarization. So again, certain stimuli can cause potassium to leak out through the channels, and that can prevent action potentials from occurring. Now, certain other stimuli can cause sodium channels to open, and if the sodium channels open, then sodium ions will flow in. When these positive charges move in, it makes the environment more positive on the inside, and that moves the membrane potential closer to zero, so it's said to be depolarizing. It's moving it away from the poles or the extremes. So here we see the sequence of events that occur. Again, if you have the potassium channels open, then they'll leak out of the cell. It becomes more negative. It's a hyperpolarization. If we have sodium channels open, then we get positive charges flowing in. When they flow in, it makes it more positive inside the membrane, and it moves closer and closer to this zero number here. Now, the magic number is at the threshold. At this threshold, then we throw open all the sodium channels, and as a result of that, you get a flood of sodium ions moving into the cell, and there's a major depolarization and actually an overshoot above zero. So you actually reverse the membrane potential inside the membrane, and you create this electrical impulse or action potential. Now, again, certain events can cause the sodium channels to open. When those open, here it's positive inside, negative out, or positive outside, negative inside. We get sodium rushing in, and that leads to the reversal of charges. You get a positive charge on the inside and a negative charge on the outside. Now, certain portions of a neuron can experience these graded potentials where you open some of the sodium channels, and that creates a more positive environment inside the neuron or inside the membrane, but it's not enough to reach threshold and open up all those sodium channels. Now, with these voltage-gated ion channels, when sufficiently stimulated, when they reach threshold, then all these sodium channels are going to open. Now, the potassium channels aren't going to open at this time. It's going to be the sodium channels that open. When the sodium channels open, again, we get this flood of sodium ions entering in the neuron, and that leads to the creation of the action potential. So action potentials are the result of this change in membrane potential due to these channels opening up. And again, the channels open up because of some sort of a stimulus that will prompt them to do so. All right, let's put our thinking caps on here. We know that, okay, at this point, at resting potential, you have some potassium channels that are open. We have relatively few or no sodium channels that are open. And we know it's got a lot of high concentration of potassium ions inside and a high concentration of sodium ions outside. So at this point, at resting potential, we're going to be leaking out some potassium. So the potassium is going to cross over, and we stay relatively negative. Also at this time, we have the sodium-potassium ion pump, and that's going to be, again, contributing to this negative membrane potential because you're pumping out 3 sodium ions for every 2 potassium that you're bringing in. Now, when stimulated, some of the sodium channels will open, and you move toward this threshold where all of the sodium channels open, and you get this rush of sodium in, reversal of membrane potential. Now we reach this point. What do you think happens with the sodium channels when it stops becoming more positive? Well, the idea is sodium channels are going to close. When the sodium channels close, then the potassium channels will open. And when the potassium channels open, again, more potassium leaks out of the cell, and that causes this repolarization that occurs. It becomes, again, more negative inside the membrane. Then you have this little undershoot here, and eventually you get back to resting potential where you get the steady leak of potassium out, and you have the pumps working, and life goes on. All right, let's see. We've talked about this. Threshold is significant because that's where the gates get thrown open, all the sodium gates get thrown open. And it's important to recognize that the action potential produced at the axon is an all or none event. Either everything gets thrown open and you get a rapid depolarization, or the signal is not strong enough, you do not get the action potential created, and poof, the signal fizzles out. Okay, the signal is not sent on. So again, when we talk about information processing, it all boils down to, will you open up enough sodium channels or enough of these voltage gated channels to cause depolarization. All right, let's see. Now, a neuron can produce hundreds of these action potentials for every second, and the stronger a stimulus may be, the more rapidly these action potentials will be produced. All right. Now, we've already spent some time looking at some of the events of an action potential, but this is a means of helping sort of visualize what occurs. So at resting potential, sodium channels are closed. Some potassium channels will be open, so you get some leakage of potassium out. Now, when a stimulus is experienced, again, some of the sodium channels, some of the sodium channels are going to open, so you get some flow in. Eventually, you hit threshold here. All the sodium channels are open. Lots of sodium falls in. You get reversal of membrane potential. And then at its peak, okay, the sodium channels are going to close off, and then the potassium channels will open up and allow the potassium to leak out. And then you'll hit this undershoot where the potassium channels haven't closed yet, and you have the membrane potential actually fall below resting potential. Once most of the potassium channels close up, then the ones that remain open will allow potassium to leak out. The sodium potassium ion pumps will work, and you're back to your resting potential. It's great to sort of talk yourself through a diagram like this if you're having trouble conceptualizing, because if you can explain it to yourself, then that puts you on a better footing to explain it on an exam. All right, well, this sort of basically takes us step by step through what we've already discussed. Again, at rest, most of the channels are going to be closed. You get some potassium channels that are open, stuff that leaks out, and it's negative on the inside. Now, with there being a stimulus, some of the sodium channels may open. If you reach threshold, then they'll all open, and you get this rapid change in membrane potential. Once you hit that peak of the action potential, then the sodium channels will close, potassium channels will open, and you get this repolarization that occurs as potassium ions flow out. When undershoot occurs, again, potassium is leaking out. Most of the potassium channels will eventually close, and then you'll have a return to this resting potential. Now, when it's all the way down here at the bottom, are you going to be able to open enough sodium gates and open several sodium gates to cause another action potential? Well, no, and that's what's referred to as a refractory period. During a refractory period, you can't open those sodium channels, and you will not be able to create another action potential. So keep in mind, this action potential and return to resting potential can take place in about 3 milliseconds. So events are quick in this process, but during the refractory period, you can't generate another action potential. Now, these potentials will move down the axon in one particular direction, starting at the hillock and then terminating in the synaptic terminal. Now, when it reaches the synaptic terminal, again, those vesicles can fuse with the membrane and dump the neurotransmitters into that cleft or space between neurons. All right, one important point to make here is that the movement of that message, the movement of the electrical impulse, again, is unidirectional. And that's because when you have the action potential, okay, you reverse the polarity inside and outside the membrane. What that will do is stimulate neighboring sodium channels to open as well, and then that will propagate or move the action potential down the axon. Well, if you remember that refractory period that we talked about, you can't stimulate another action potential in the other direction. So what that means is one neighbor can stimulate another neighbor, but it only goes down the axon. Because of this refractory period, the impulse can't be sent back toward the neuronal body, which is great, right? Because you want to be sending the message out. You don't want the message to revert back and potentially interfere with subsequent messages or impulses. Now, one way to speed this process up is through the glial cells, in this instance referred to as Schwann cells, that produce myelin. Now, myelin is this fatty coating that surrounds axons. Now, if you think about an electrical wire, if you've got an extension cord, that electrical wire is wrapped in insulating cloth and rubber and plastic to prevent, electrons from crossing over from one wire to the next. If you have a couple of bare wires, then you can short circuit, whatever it is you're trying to plug into the wall. Well, if you have billions of neurons traversing your nervous system and they have to send impulses at highly precise times, you can't have neurons crossing over one another and stimulating the axons unnecessarily. You need the impulse to transfer information down to the synapses. So you have Schwann cells produce this myelin, this fatty myelin coating that protects the axon and prevents messages from crossing over into neighboring axons unwanted. Let's see. Now, the Schwann cells, as we said, are glial cells. They're those support cells that help the neuron conduct its job. Now, another important location here are called the nodes of Ranvier. How romantic, right? But the nodes of Ranvier are where the action potentials actually occur. These are bare spots that don't have the myelin coating. All right. Let's see. Now, this process of sort of skipping over is referred to as saltatory conduction. The salt and saltatory here referencing the sodium ions. Now, the way I conceptualize this is I think of going to like a football game, go to Lucas Oil, wherever the case may be, and people want to perform the wave. If you're sitting in the stadium and people are performing the wave, you might have to wait a long time as the wave passes along the crowd before it will loop back to you, right? Well, imagine at the stadium there are huge sections that are blocked off and people aren't sitting there. So if you begin the wave in one section, well, at the section that's blocked off, the wave will then jump to the next section of people. You go to an area that's blocked off and then it jumps to the next section of people. This will actually now happen faster. So by myelinating the axon, you allow the action potential to be sent, but the action potential can actually skip over this myelin coating, speeding it up. So you don't have to wait for channel to open as it progresses down. You can actually open channels further down the line. It's obvious, you can't have channels opening when you have this fatty coating, so you can't have that ion passage through there. Now, demyelinating diseases like multiple sclerosis will actually have your body attack the myelin surrounding your axons, and that causes degeneration of nervous impulses. So in this case, the motor neurons that supply the muscles have their signals degraded, and as a result, people will experience weakness and difficulty with coordination and things like that as a result of their demyelinating disease. All right, we'll move on to the next section here in just a bit.